Without further ado, we'll have our first speaker up today. I'd like to introduce Ishrat Zaman. Ishrat Zaman is a scientist working in the field of medical research as a bioimage analyst. She is passionate about the intersection of neuroscience, image analysis, and open source software. Today, she is going to talk to us about how you can reproducibly identify and count individual neurons in a tiny, diffuse brain region surrounded by lookalikes, and when each 3D brain image is multiple terabytes in size. Please welcome her to the stage to present Big Brains, Small Targets, Whole Brain Imaging with Python.
Yay, okay. All right. Hi, everyone. Thank you so much for the very kind introduction, and thank you for having me here today. I'm Ishrat. I'm a bioimage analyst at WeHi, which is a medical research institute, and today I'll be talking a bit about a project we've been working on for the last few months. This talk is a bit more of, as Genevieve mentioned, a bit of an application of all the cool Python tools that we've used and how we've kind of strung the different packages together to be able to analyze the type of data that we have been. So the project I've been working on for the last few months, just to give everyone a bit of a background for those of you that aren't from a biology background or medical biology, this is a mouse brain. So this is what a mouse brain looks like. And essentially that's image from the top down looking this way, essentially. And if we cut through the brain like horizontally this way, this is what the sections look like here. And I'm going to refer to them as coronal sections, just to set that up. And essentially, the researchers that I've been working in have had this overarching goal of counting the number of neurons in one specific region of the brain. Now, this sounds easy enough, but when you realize that this is all of the different regions in a mouse brain, if I'm zooming through coronally, you can see there's a lot of regions, and they're all quite small. I don't know how you'd be able to tell one apart from the other. There's some big ones, some small ones. There's a lot of regions. And the region that the researchers are interested in is one related to Parkinson's disease. So they were interested in this small region here that I've outlined in white, and this is called the substantia nigra pars compacta region, or the SNpc or SNC. And they're interested in this region because this area is densely packed with dopaminergic neurons, or dopamine-producing neurons. And in the context of Parkinson's disease, there's significant neuron loss in this area, and accurately understanding the neuron loss in this SNpc region is key to understanding disease progression, as neuron loss is what drives some of the symptoms and effects of Parkinson's disease. And so with the advancement of sample preparation techniques and microscopy techniques as well, the researchers are able to take the brain, which looks like this, and clear it using a process called tissue clearing, which essentially makes the entire tissue transparent. Then they stain or label the tissue with a marker called TH, or tyrosine hydroxylase, and this basically attaches directly to these dopamine-producing neurons and attaches a fluorophore to it, so we'll be able to see it when they image it using the light sheet microscope. And the light sheet microscope is really, really cool because it allows you to image entire organs rather than thin sections, which is what most microscopes normally do. And so the benefits of this is that we image the whole brain, so we're also imaging every single positive cell. And hopefully we'd be able to automate the analysis so that we can get the most accurate counts, ideally. And so just to show you what the images look like when they come straight off the microscope, this is what it looks like if I'm zooming through in the orientation that it was acquired, and this is the autofluorescence channel, which I'll talk about in a second. And then this is in the TH channel now, where it starts to zoom through, and this is the channel that's labeling all those dopamine neurons. And you can see when I zoom in here, all the little positive dots are all those individual neurons there. And just for a bit more context, each of the pixel size or voxel size is roughly about 2 micron in X, 2 micron in Y, and 7 micron in Z. And so they're really, really small microns, and especially when you consider that this is roughly a 1 centimeter cubed volume in total. And now if you want to see what it looks like in 3D, we've got the video here where it's rotating around. And this is the autofluorescence channel, which I mentioned earlier, which is essentially just the natural fluorescence of the tissue. And you can see this channel holds a lot of information about the structure and architecture of the brain. And now this is the TH channel in 3D, and then I'll zoom into that region with those positive cells, and you can see there's a bunch of those little positive cells there. And so as I mentioned, the pixel size and the size of the tissue, you can imagine that these data sets are quite big. So each of the data sets is 2 terabytes, roughly. And of course, this is science, so you can't just have one brain. Between the researchers I've been working with, the initial batch had 20 brains, so we had roughly about 40 terabytes of data to analyze. And so, as I mentioned, the autofluorescence channel holds a lot of information about the architecture. But I'm not a neuroanatomist. I'm not from a neuroscience background necessarily, but you know, we can see some of the shapes, but I couldn't tell you what region is what. I don't know what the different regions are. If you tell me to extract a particular region, I couldn't tell you where it is. So basically, the first step is to register our raw data to a reference atlas. And so this reference atlas is one from the Allen Institute, and this is a really, really thorough, really, really in-depth atlas that was developed with a lot of effort. But basically what we want to do is align our raw data to the reference atlas, so hopefully we'll be able to know what regions we're looking at. And of course, this is where we begin to use Python. So the first package or first tool we used is one called BrainGlobe. And this was a really cool Python ecosystem that was developed by the Neuroinformatics unit at the Sainsbury Wellcome Centre. And it's really great because it holds a lot of tools for analysing whole brain data sets, from brainreg or BrainGlobe registration, which registers your data sets, to CellFinder, which is a cell detection tool, and also BrainGlobe segment, which can segment out different structures of the brain. And all of these tools have a Nipari plugin, and Nipari is also another open source Python tool, which is really, really great for viewing multidimensional data, particularly images, and it's used a lot in bioimage analysis. And so here I'll show you, this is what the Nipari plugin looks like. So it's really, really great. It's got a great user interface where we can just load in our raw data, fill in some metadata about the brain, and also there's a bunch of parameters here that you can see here that you can change as well. So the main ones I'll kind of mention is bending, energy weight, grid spacing, and smoothing, because I think these are the ones that can have quite a big effect on the registration. And since this is all in Python, it all integrates really nicely, and we can scale up all our code as well. So yeah, once we load it in, we can run the registration here, and then this is what the registration looks like. So I'm just going to zoom through our data in the coronal orientation, so you can see this is what our brain looks like. You can see some of those structures coming out quite clearly. And this is just the autofluorescence channel now. And then if I overlay now the atlas on top of it, so now you can see this is how it's been registered, and now this is telling us where all the different regions are within our brain. And I've just used Nipari to visualize this, so you can see it looks pretty good. It's pulling out some of those big structures. And now if you want to see it in 3D, because who doesn't like 3D videos, you can see there's the data set spinning around with the atlas overlaid on top. So now we know what the regions are in our data set. And I can pull out the specific SNpc region now, which I show you here. This is what, after alignment, it's being called as the SNpc, or the region that we're interested in. And then I'll switch over to the TH channel in a second, which will show you where the positive cells are as well. I think that'll start now. So you can see all those little bright cells popping up directly within that region there. And so now that we have this, we can start using a few more packages, you know, SciPy, NumPy, scikit-image, dask-image particularly, and pandas to take our region that's been extracted, put it into the original orientation that the data was acquired. We can pull it back up to full resolution. And now we have the region in the context of the entire brain. However, this region is still, this entire brain is 2 terabytes, you know, and analysing this would be an absolute beast. But because we're really only interested in this region here, we can just narrow down the region there and just run our analysis on that smaller region, which pulls it down to less than a gigabyte, which is much more manageable. And so now that we have this region and we have the full resolution data, we can use Cellpose, which is a deep learning cell segmentation tool. And I can show you here, this is what the region after we've pulled it out looks like. You can see all the positive cells popping up here. And then Pradeep, who's another member of our team, developed a custom-trained Cellpose model to detect all of the cells in this region here. So when I start going backwards, you'll be able to see all the different cells it's detecting popping up. And it was a really great, Cellpose is really great. It just works really, really well, and it's really easy to use to train your own model. So now you can see all those different individual cells popping up as the cells that we've detected. So now just to summarize what our initial analysis pipeline looked like. So we first registered and aligned our data to a reference atlas using BrainReg and Napari, so we know what regions of the brain we're looking at. Next, we wanted to, when we were analysing the rest of the data set, we wanted to run a parameter sweep on 2 or 3 just to see if we could find what parameter combinations seemed to work well between 2 or 3 data sets. And then once we found that, then we could apply that set of parameters to the rest of the brains using all of these packages, and we could extract out the SNpc region. And then finally, we would use the custom-trained Cellpose model to count the Th-positive neurons in the SNpc. So once we do this, we have this sort of distribution of all the different counts for all the different samples. So we should be done, right? Well, no, absolutely not. So the problem turned up when we were kind of going a bit deeper into the results and sharing that with our collaborators. So when we applied the registration, again, not a neuroanatomist, but all I have to go off is what the boundaries look like on the data. And so, the registration looks pretty good. It looks like the boundaries are quite tight on the tissue. Some of these big structures look like it's aligned pretty correctly. And then when we look at the region here, we can see, okay, it looks like it's capturing most of those cells. But when we first started analysing it, we didn't know, if these cells were also meant to be in the region, or if these cells, or what about all of these? So, we were asking the question of have we just completely missed the region? Well, the answer is actually, apparently, all of the surrounding regions around the SNpc are also expected to have positive cells, which makes our lives a lot more difficult because now we don't know where the boundary begins and where the boundary ends for one region to another. And then further, when we use the exact same parameters on a different data set, you can see, again, globally, the registration looks really good. We can see it looks pretty similar to the last one. But then here, when you look locally in that SNpc region, it looks a bit like maybe it's shifted down a little bit compared to the last one, and maybe it should be shifted up a little bit more. But then for the third brain, again, globally, exact same parameters, it looks good, but it looks like it's completely missed that region there. Which makes things a little bit difficult because now we don't know which one's correct and if there's any problems, we don't know why there's this inconsistency. So further, the channel that's used for registration is I noticed that when I run the registration and I pull out this region here, it looks pretty good, but the cell counts say roughly about 3,000 for this one. But then if I change even one of the parameters on the exact same data set, the region shifts just a little bit, and again, we don't know which one's right or wrong. Both of them look pretty good, but suddenly the counts have shifted by 500. And so now this makes our life quite difficult because we don't know which one's correct, and the researchers are also expecting subtle changes in the counts of neurons in these regions. So if the counts can change this much just by our registration, then there's a bit more of a problem and we need to look into it a bit deeper. And also, just to highlight again, this is just a small section from the autofluorescence channel, but you can see, maybe there are some structures. There's some big ones here, this one here, but apparently these are all the regions that are in this slice. So what makes it difficult is that you can't see the boundaries easily in the image. And this is the SNpc region, this light green one, but I don't know where that begins or where that ends at all. So just to summarize some of the challenges in registration, well, firstly, we could have inconsistent signal where the data sets look different from one another, and that could cause problems downstream. Next, there's ambiguous borders. There's no obvious outline to the region in the channel that's used for registration, or in the TH channel either, so we don't know where that is. We also don't have a ground truth, so you can see these are two different regions that it's pulled out with slightly different parameters. Both of them could be correct. And also, if we misregister, then we could overshoot the region entirely, which should be here, but it's being called as being here. Now, why does this all matter? Well, subtle misalignments in a small region like the SNpc, which is surrounded by other regions that also contain Th-positive cells, can lead to incorrect cell counts, which will ultimately result in misleading biological conclusions. So we thought, okay, well, we need a ground truth. We need an answer. We need something to compare against. So we took 3 of the datasets, we passed them to our collaborators, and they used Cibra, which is another really cool Python-based tool to kind of explore atlases in all the different orientations, so that was really helpful for them. And they also used the Allen Atlas to help them manually annotate 5 to 10 slices per brain. And this meant that I had a set of ROIs from them, and I could interpolate the ROIs, and that meant I would have like essentially a 3D ground truth region. However, I just want to note some caveats. Well, so these annotations are based on the Allen Atlas, which is built on thin sections, and it's not built on a tissue that has undergone tissue clearing process, which can cause some tissue deformations. Also, the annotators struggled to identify the regions themselves. We didn't get them to do the same one, but they struggled to know where the boundaries are. But nonetheless, it was a good starting reference for us. So now that we had a point of comparison, we can get F1 scores, which is essentially a metric of understanding how well a registration has performed between a ground truth and the registration. We wanted to know, are there a set of parameters that actually work consistently for all the different data sets, so that we can hopefully be confident for the data sets that don't have a manual annotation or a ground truth. So we thought, let's just run a parameter sweep and just see how the regions and counts change, and maybe the final output might actually be a distribution of counts rather than a final single count. So our initial parameter sweep involved changing those parameters I mentioned earlier, and we had roughly about 150 combinations of these 3 parameters per sample that had the annotation. And then I submitted these as an array job using Slurm for some parallel processing, which made things a lot faster. And then we assessed the F1 scores to identify a set of stable parameters across all the datasets. And spoiler alert, no single parameter combination worked consistently across every dataset. And just to show you what this kind of looks like, so the red is what was annotated as the ground truth, and the blue or purple is kind of what it looks like, what the different parameters are calling as the SNpc region. So you can see the region shifts around and jumps around quite a bit. And also I'll just highlight the blue region looks like that, a little bit different to the ground truth because this was used using an atlas that was developed specifically for tissue cleared images. And you can see, yeah, there's quite a lot of variability. And so to show you what the final output looked like as this distribution of counts, you can see there's quite a lot of variability. And particularly there's some where the F1, so I've got the counts here and the F1 scores here, and you can see there are some that completely miss the region entirely. And then I've got some of the videos here where you can see that happen, where the regions just completely shoot off. And so I noticed this one here, 324, and I noticed there was so much variability in between different parameter combinations, and I thought maybe that's because the image itself has quite low signal-to-noise ratio. So I thought maybe we have to do some pre-processing. So using dask, NumPy, simple ITK, and pandas, I essentially wanted to take the raw image and make it look as much like the reference atlas as possible, because I thought if I could do this, then hopefully the alignment would work a lot better. So first we did an N4 bias field correction using simple ITK, which essentially normalizes the intensity. So you can see there's this really bright region in the center of the raw image. However, after this correction, that's gone and it's a lot more uniform. I then histogram matched, so I matched the intensities of this image to the reference atlas, and then I removed the background. So now the image looks a lot more similar to the reference atlas. And then just to show you a little plot to summarize this was a big data set, a big group of plots, but I've just zoomed in on one. So this is the F1 scores where the grid parameter was set to this, and then all the different combinations of parameters down there. And just to show you can see the F1 score jumps around quite a lot on the unprocessed or raw data, and particularly for that 324 one that had the really low signal-to-noise ratio. However, after pre-processing, suddenly all the F1 scores came up, and they were much more uniform between each other. So now 324 was a lot more comparable to the other samples, and also, no matter what parameter combination I chose, the registration was a lot more consistent, which seemed like a win for us. And then now, after running the registration on the pre-processed data, you can see the counts have tightened up a lot, and there are no registrations that completely overshoot the region here, which was a really good thing for us. And you can see that down in the videos down here as well, that the registration isn't as variable. And just to show you side by side, the cell counts on the raw data versus the pre-processed data, and then the same for the F1 scores here. So now we had a distribution of counts rather than the single counts that we had at the start. However, we thought instead of picking one parameter set or having all of these different ones, we thought, what if we combine them all? So we built these consensus maps where we overlaid regions from the different parameters, and so it gave us this kind of distribution of where all the different regions are, and I did this using NumPy. And so you can see here the light blue is what was annotated as the ground truth, and you can see but again, I just do want to highlight the ground truth. There is also a lot of uncertainty in that ground truth itself, but you can see all the different, the regions are sort of getting us closer to what we think is the truth. And this just gave us a way to kind of embrace the uncertainty rather than ignoring it. And just to show you what that looks like here, so at a threshold of, say, 0, where this is essentially a combination of all the masks, all the different registration parameters, and then, say, t equals 0.5 or t equals 1. And essentially, this gave us not just one number, but kind of a range of plausible neuron counts. And you can see here, I've just plotted all the different counts for all the different consensus thresholds. And you can see that it all kind of follows the same pattern, which I guess is a good thing, but you can see there's a bit more of a stable region within here. So this would probably be where we could report our sort of final counts. And, you know, the truth is that even though experts can't tell us exactly where the SNpc region is, the consensus maps and pre-processing kind of brings us closer to something that is a little bit more reproducible and ideally interpretable. So it seemed like a solution for us for when we don't really have a ground truth. So to summarize our current analysis pipeline, we basically read in the data using dask image, which is really great because data is huge, so this gives us a way to read in the data efficiently rather than loading in 1 terabyte of data into memory. Pre-processing, which I use simple ITK, NumPy, and pandas. Doing the registration parameter sweep using BrainGlobe, brainreg, Nopari, and scaling everything through Slurm. Identifying consensus regions using NumPy. Extracting out the SNpc regions using all the tools, including scikit-image and SciPy, and also running the cell detection at the end using Cellpose and Slurm. So essentially, all of this was all wrapped into a pipeline that scales on our HPC clusters, which allowed us to analyze the 40 terabytes of data, and we've got another 40 terabytes incoming soon. So just to summarize the conclusions and key takeaways, so accurate registration of small and diffuse regions is hard, but pre-processing seems to help, and the consensus region helps us tackle this uncertainty. However, this really is still an open question, and I would be so open to getting any advice or thoughts or comments from you all. And most importantly, Python is helping make neuroimaging analysis reproducible and also scalable. So it's given us a really great way to analyze these types of data sets that we might not have been able to analyze in other ways. And just finally, I'd just like to thank everyone, particularly my team and the Centre for Dynamic Imaging at WeHi, especially my bioimage analysis core team, Lockie, Pradeep and Mike, and also our collaborators that gave us all of this really great data to work with, and also, of course, the world of open source and all of the awesome Python packages and tools that we've been able to use. My email's there and my GitHub's there as well if you'd like to reach out to me. So thank you.